What does CRISPR stand for? CRISPR stands for C. Clustered, R. Regularly, I. Interspaced, S. Short, P. Palindromic, R. Repeats. Here's a video just to give you a context about CRISPR and gene editing. This is a DNA line segment, and this is a Cas protein. It chops up DNA like molecule scissor. When a virus invades a bacterial, Cas protein cuts out a segment of the viral DNA to stitch into the bacterial CRISPR region, capturing a chemical snapshot of the infection. Then those viral codes are copied into short pieces of RNAs and then sent into the lab. Who discovered CRISPR-Cas9? Biochemist Jennifer Duane and microbiologist Emmanuel Charpentier co-invented the gene editing system CRISPR-Cas9, a technology for editing DNA with precision and efficiency. Where do CRISPRs come from? CRISPRs were first discovered in archaea and later in bacteria by Francisco Mojica, a scientist at the University of Alicante in Spain. He proposed that CRISPRs serve as part of the bacterial immune system, defending against invading viruses. They consist of repeating sequences of genetic code interrupted by spacer sequences, remnants of genetic code from past invaders. The system serves as a genetic memory that helps the cell detect and destroy invaders called bacteriophage when they return. Mojica's theory was experimentally demonstrated in 2017 by a team of scientists led by Philip Horvath. In January 2013, the Zhang lab published the first method to engineer CRISPR to edit the genome in mouse and human cells. What's the use of CRISPR? Uses of CRISPR. CRISPR genome editing allows scientists to quickly create cell and animal models which researchers can use to accelerate research into disease, such as cancer and mental illness. In addition, CRISPR is now being developed as rapid diagnostic. These repeats are small pieces of virus found in the DNA of some bacteria. The bacteria incorporate these small pieces of DNA from viruses when the viruses invade the bacteria. CRISPR helps prevent disease like HIV and cancer, make crops more hardy, and tackle the climate crisis. Okay, how does the system work? CRISPR spacer sequences are transcribed into short RNA sequences, CRISPR RNAs or cRRNAs, capable of guiding the system to matching sequences of DNA. When the target DNA is found, Cas9, one of the eczema product by the CRISPR system, binds to the DNA and cuts it, shutting the target gene off. Use modified version of Cas9. Researchers can activate gene expression instead of cutting the DNA. These techniques allow researchers to study the gene's function. What other scientific uses might CRISPR have beyond genome editing? CRISPR genome editing allows scientists to quickly create cell and animal models which researchers can use to accelerate research into disease such as cancer and mental illness. In addition, CRISPR is now being developed as a rapid diagnostic, as I mentioned that before. To help encourage this type of research worldwide, Feng Zhang and his team have trained thousands of researchers in the use of CRISPR genome editing technology through direct education and by sharing more than 40,000 CRISPR components with academic laboratories around the world. The video I used was from TED Talk, and for the research, I researched from Broad Institute. Thank you for sitting throughout the video.